My name is Silas, and I'm a PhD student at the Niels Bohr Institute in Copenhagen. Have a look at this image and note the diversity in the species. I would like to ask you, what do all of these species have in common? And probably what would come to your mind is that they all build on the coding sequence of life on Earth, DNA. However, during this talk, I would like to focus on two other shared properties, two directions. They all have an inside-out direction, and most, if not all, consist of a set of organs with a specific direction of the internal flow. With those two properties in mind, I will see how far I can get in building a complex organism in silico. Let's start at the very beginning. Have a look at this image. This is the mouse morula 3 days after fertilization. And note the complexity in the gene expression markers. 1.5 days later, it had developed into what's known as the blastocyst, where the same gene expression markers now form 3 distinct cell lineages. In the PLOS biology paper, we asked, how does this process occur? What makes it so successful? And how can it be that it's so robust in morphology and so similar across the mammalian species? In order to answer questions like these, we constructed an agent-based model consisting of a minimal number of developmental rules. So what you see on this slide at the bottom are immunostaining images similar to the ones in the previous slide. And on top is a diagram showing the development from fertilization to implantation. Right? And here you have the 4 rules in the agent-based model. Right? So I don't have time today to go into detail in each of the rules. However, I do want to emphasize the first rule, the polarity rule, which we apply on day 3. This rule enables us to make an inside cavity, the blastocyst, and thereby making a stable shell of cells that can then grow and expand and later they become, in this case, the blastocyst. Let's see the model in action. Here you see a 2-dimensional example. And we also did three-dimensional simulations in the paper, and we use this for making a prediction, experimental predictions that we tested and confirmed experimentally, which you can find in the PLOS biology paper. All right, but for now, I just want to recap and have this focus on cell polarity. All right, so it seems like, going a little bit quick here, that the embryo utilizes a minimal versatile toolbox. We made this agent-based model consisting of 4 rules, and here now I list the components in these 4 rules. Right? And what we're suggesting here is that this is sufficient to make the blastocyst. And then you can imagine that you could capture a number of other developmental transitions and stages, possibly by just adding 1 or 2 more components. Let me dig a little bit into the first rule. That's like what enabled us to make the cavity. And now I would like to go a little bit more into detail about how did we implement this rule, and how do we implement polarity. And before I do that, let me ask a few more questions. How do complex shapes form in general, not only the blastocyst, and how are they maintained in time? All right, so think about your internal organs and then ask, how can it be that we see so diverse morphologies? And you could think about the growing heart, and then we asked, what makes this so robust to cell growth, to proliferation? All right, so in order to answer questions like these, and that's also how we implemented cell polarity in the blastocyst example, we developed a very simple model that I would like to teach you quickly now. It's so simple. All right, so think about this as being 5 cells. Right, and they could have any shape and they could be positioned anywhere in this 2-dimensional example. The cells, they could have some kind of gradient or somehow a polarity, here shown by the yellow to red gradient. And what we then do is that we represent each of the cells by their center of mass in blue, and the gradient here pointing towards yellow by an arrow. Right? And that's basically the entire model. That's these point particles with spin, nothing else. We neglect the cell shapes. And then we add a couple of equations of motions, a soft potential, a soft repulsive force, and an attraction term. And this attraction term, that consists of an attraction factor, here called S1, which depends on the spins, the directions of the spin, and the displacement vector between two cells, Rij. Right, and this is maximum, this factor is maximum when the two cells, they line up and look by the arrows, when the arrows point in the same direction. That's where the factor S is 1, and it would be -1 in case that they look in opposite directions. It would be 0 if the two spins, they are perpendicular to each other. Then we just sum over all your neighbors, and we can add some noise. Noise is not important for the results. The result is, as long as you don't turn up the noise too much, then you will get qualitatively speaking the same results as I'm presenting for the rest of the talk. Okay, but let this one, and then you find the minimal energy configuration, which is in 2 dimensions would be aligned with the arrows looking in the same direction. And this is our very simple epithelium, right? But now you can just expand this. You can expand it easily to 3 dimensions, and you can easily expand this to 10,000s of particles, 10,000 of the cells. And that's what I did here. This is an aggregate of, I think in this case, there are 8,000 particles. And now, initially, the polarities is shown by blue to yellow, or deeper inside the structure, more dark blue to red, is pointing initially in all kinds of different random stochastic directions. But now just let it develop. But just apply these equations of motions, and it will develop, it will expand, it will start to make internal lumens and sheets that will fold until it reaches a final state, a minimum energy configuration, a metastable topology where it will stay forever, basically. Right. So this reminds us a little bit about, here we're thinking about the question I raised about how can we see so rich diversity in our internal organs. Here you get all kinds of shapes that's only depending on the initial conditions, or possibly, but I didn't show that here, but you can find that in the eLife paper. You could have some external constraints that's applied at different time points, and then you can drive it towards a certain morphology. And it's not only stable in time, it's also stable to cell growth, to proliferation. He's shown by taking the number of cells and let it grow to 3 times the number of cells. And then that's remind us back to the growing heart that I showed earlier. However, I admit, right, so far we just considered some alien organs, but now I would like to show is 2 applications. So I already actually showed 1 application. I showed you the blastocyst, but let me dig into another example, another application, and ask the question, how do folded organoids form? All right, so we, together with our collaborators, got a paper out a couple of years ago where they studied pancreatic organoids. And in one medium, they find these organoids to grow spherically all the time. Right? It's just like an expanding, that's what you see on the left, just an expanding organoid with a cavity inside. However, in a different medium, that's what you see on the right, there they find that it may start, or it does start, like a small sphere, but after a few days, it starts to boggle and start to branch and make this very folded structure that you see on the right. And then we asked, is the difference in these 2 medium and the difference in the shape of the organoids, is that due to that the proliferation rate is different in the 2 different medium? Or is it due to that the pressure and the stiffness from the material makes the organoids fold and thereby drive the folding. And I admit these two hypotheses, those are not mutually exclusive, but the model allows us to explore the extremes. So that's what I will do now, right? So here you have like, basically, this is basically the blastocyst without the inner cell mass. This is just a hollow sphere of cells. We let it grow and you let it grow quickly and then it starts to buckle at some point and you get a very folded structure. And then you can start to like measure how folded this is, and that's basically what we did here. So with the way that we defined foldiness is by counting the number of local minima relative to the center of mass. And then I made a plot here where we plot this measure as a function of the proliferation rate. And then we go in, we let it grow up to a certain number of cells, and then there we measure how many local minima do we find. And that's what I've shown here for 3 different proliferation rates, or if you follow the blue dots, then it's 5 different proliferation rates. And the faster it grows, the more folded it will be at a specific number of cells. This might not be that surprising, but let's follow a little bit more. If you let this grow really slowly, that's what you see at the bottom left corner, then you will just keep on. You'll always have time to reach steady state and just make an ever-expanding lumen of cells encapsulated. And now take the one that does not fold. That's the one at the down left corner. And put that into a stiff material, simulated material. That's what I did now on the right-hand side. And then let it grow until it reaches the same number of cells, but in different degrees under different degrees of pressure. Now we find that this does also drive folding, but the folds in these cases are very different. This makes very few, very deep folds. And it's not only very few folds, it's like a saturating number of folds on the order of 8 to 10 folds, which is very different from the extreme proliferation-driven case, which made an ever-growing number of folds that's very small and shallow. So basically here we have this prediction that at least in these 2 extreme cases will give rise to very different fold morphologies. Okay, let me show you a different application. And here we're thinking about all our internal tubes. That could be the intestines, the vascular system, etc. And then we're asking, how do they get a specific thickness, or in general also a specific length? Right, and we thought about that based on a paper that came out a few years ago from my current collaborators, Stanford. They studied a planar cell polarity, and in the renal tubes, they measured their thickness. So here in blue, you see the thickness of the wild type tubes, the diameter. However, in red, you find one planar cell polarity mutant, loop tail mutant. And there they found that this mutant has given rise to thicker tubes. And that's also what you find in the images. The top image is the wild type, the bottom image is this PCP mutant. In the paper, they did several other PCP mutants and found similar results. And now we're curious about, can this simple model capture this phenomenon? Okay, but so far I only really talked about what's known as apical-basal polarity, the inside out. So let me just recap the model for you. That's what you see here. That's like the point particles with the spins, and then the equations of motions. However, now, in order to capture this phenomenon, I will have to add planar cell polarity. And that would just be like adding a second spin to all of the point particles, now shown in green. I mean, it could be any direction in principle speaking. And I will then have to expand the equations of motions to incorporate this spin as well. So everything here is symmetrical. I added a few more terms in the attraction factor, and each of these S factors, the S1 is the one that I showed on the previous slide, right? And now I just add the green spin to the planar cell polarity, and it's implemented completely symmetrical. And the only parameters here, that's the coupling constants between the different attraction factors, the lambdas 1, 2, and 3. Let me run this. So this here would be minimum when the two spins stay perpendicular to each other, when you form the inside out, the blue red, and when the green arrows point in the same direction. So that's this would basically, in this case, be the left-right, define the left-right direction. And that reminds us about like a very simplistic speaking and an arm with an inside out and a way to distinguish the fingers from the shoulders. And I mentioned the parameters here, and I pinpoint those out up there. If you sum those up to 1, require you sum those up to 1, you're basically left with 2 parameters. Okay, but now I only did like this for 5 cells. You could expand this to like thousands of cells in 3 dimensions. And that's what I did here. So this is our sphere again, our hollow sphere of cells with apical-basal polarity, but now we just add planar cell polarity to the cells. And you can do that initially in all kinds of directions, completely stochastic. The minimum energy configuration would be where the PCP curls around the sphere. And then if the third coupling constant is strong enough or high, then it will start to elongate the sphere and thereby make a tube. And it will do that until it reaches a final length and width. There's no cell proliferation here, right? And this thickness is purely defined by the degree, the value of a lambda 3, which is basically the strength of the planar cell polarity. So what I show you in this diagram is the length and the width of the tubes at different degrees of planar cell polarity. And that takes us back to the experiments that I told you about from the Renal tubes, that the wild type gave very thin tubes, whereas the PCP mutant gave rise to thicker tubes. Okay, so with this, I would like to come to a conclusion and just recap the model very quickly, maybe the potential of the model. Right, so basically what you see here is a diagram with 2 symmetry breaking events. You can start with an aggregate of non-polarized cells on the left-hand side. And then you can have the first symmetry breaking event. And that's basically what happens in the blastocyst. You go from a morula to a blastocyst, right? In that case, it's not all of the cells that break their symmetry. And therefore, you still have like an aggregate of cells inside the cyst, and that's known as the inner cell mass. However, what I show you here is like a more general perspective on it. And this is basically lumen formation in general. Then you could later on have a second symmetry breaking event, which will then be by adding planar cell polarity, and that will start to make tubes. And you could go back and forth here. Cells could lose polarity, and you could apply this to fractions of cells and so forth. And then you can imagine that you could get all kinds of different complex, stable morphologies. And that's basically taking me to the conclusion. All right, I showed you the blastocyst, where I suggested this, the minimal versatile toolbox. And then I pinpointed out the cell polarity rule, the first rule in that model, and then I showed how that enables us to make diverse and robust shapes. Here I showed 2 3D prints in gypsum, one colored, another one not colored, of the model. The one on the right hand side, that's the alien organ that I showed in the very beginning, and the one on the left hand side, that's basically similar to the organolds folding that I've been showing. And with this, I just want to acknowledge all the people behind these studies, my co-authors from the Niels Bohr Institute and from the Danish Stem Cell Institute in Copenhagen. Thank you for listening.